Blood, it's a special connective tissue. It's mesodermal in origin. Study of blood is hematology. William Harvey, father of blood circulation, as he demonstrated the blood circulation in living snail. Healthy males have 5 to 6 liters and females have 4 to 5 liters of blood. Mollusks have blue color blood due to presence of copper containing hemocyanin pigment. Blood consists of plasma and formed elements. Plasma is also known as fluid matrix. So when we see the plasma here, it's straw colored viscous fluid and it constitutes of 55% of the blood. Now if you see the plasma contains 90 to 92% of water and rest 6 to 8% are the proteins, that is fibrinogen, globulins and albumins. So these are the proteins. So what all are present in the plasma? Water 92%, some ions, proteins that are fibrinogen, globulin and albumins, and apart from some nutrients, some wastes and gases. So fibrinogen, which is present in the plasma, they are needed for clotting or the coagulation of the blood. Globulin, these are primarily involved in the defense mechanism of the body, and albumins help in balancing the osmotic balance. Okay, so plasma also contains small amount of minerals like sodium ion, calcium ion, magnesium ion, bicarbonate ion and chloride ion. Glucose, amino acids, lipids are also present in the plasma as they always in transit in the body. So that means they are always moving from one place to other place as per the body requirement. Plasma without the clotting factor is known as serum. Now secondly, the formed elements. Formed elements, if you see, there are 3 types of formed elements. These are erythrocytes, leukocytes and platelets. Now formed elements constitute 45% of the blood. Firstly, we will see the first formed element, that is erythrocyte. So erythrocytes are also known as red blood cells. Now these are the most abundant of all the cells in the body. Okay, a healthy adult man on an average has 5 million to 5.5 million of RBC per cubic millimeter of the blood. Now RBCs, these are formed in the red bone marrow in the adult. These are devoid of the nucleus. So the nucleus is not present in the RBC in most of the mammals. And the shape is biconcave. Here you can see. So they have red colored iron containing complex protein called as hemoglobin. Hence the color of the name is given to these cells, erythrocytes. Now a healthy individual have 12 to 16 gram of hemoglobin in every 100 ml of blood. Hemoglobin molecule play a significant role in the transport of respiratory gases. RBC have an average lifespan of 120 days, after which they are destroyed in the spleen. So that is why the spleen is also known as the graveyard of RBCs. And if you see the sizes, the smallest RBC is present in the musk deer and the largest RBC is present in elephant. Okay. So the next will come leukocytes. So leukocyte, what they are doing, these are also called as the WBCs or the white blood cells. They are colorless because of the lack of hemoglobin. They are nucleated, that means nucleus is present. Now they are relatively lesser in number and the number is 6,000 to 8,000 per millimeter cube of the blood. And these are generally very short-lived, like RBCs were 120 days, but these are short-lived. Now, 2 main categories of WBC are there, and the categorization is done on the basis of presence or absence of the enzyme-rich specific lysosomal granules. So these are the granules on the presence or absence that has made the basis for their categorization. So granulocytes, if granules are present, granulocytes, if these granules are absent. So neutrophil, eosinophil and basophils, these are the different granulocyte, while lymphocyte and monocytes, these are the agranulocyte. So this can be MLA, that means monocytes, lymphocytes are agranulocyte. sites. So they are not containing the pigment, that granules. Now coming to the leukocyte. Firstly, we will see the leukocytes. What are the different type of the leukocytes present? So first, neutrophils. So neutrophils, these are most abundant cells, that is 60 to 65% of the total WBCs. These are phagocytic cells, that means they destroy the foreign organism entering the body. And these cell, if you see this staining response, so they can respond, these cells can take up both acidic and basic stain equally. That is why they are called as neutrophils. Okay, and these are also called as the micropolicemen, okay, of the blood. So that is why these are also very important. Coming to the second, basophils. These are least in number, 0.5 to 1%. They are on the basis of their name, basophils. These are base loving. So they appear blue in color. So on the basis of the stain response. Now these cells take up the blue stain. Now the function, that is, they mediate the inflammation. How they mediate inflammation? By secreting histamine, serotonin, heparin. So that is why they are involved in the inflammatory reactions. Okay, and these are least abundant. Third is the eosinophil or acidophil. So they are acid loving, means they will appear red in the color after staining. So cell will take up red stain. Now what is their percentage? They are 2 to 3% of total WBC. Now they will resist infections, and they are also associated with the allergic reaction. Okay, and they secrete the histamine also. They are also very effective against the large sized parasite, and they have the bilobed nucleus also. Like here you can see bilobed nucleus is there. So all these are the granulocyte. Now coming to a granulocyte, that is monocyte. Now monocytes, they have 6 to 8% of the total composition of the blood, WBC. Now phagocytic cells, they will destroy the foreign organism entering the body. Okay, so they will transform into the macrophages, and these are the phagocytic cell macrophages. They will enlarge, enlarge to form macrophage, and so they have very important role in the antigen presenting cell also. Now lymphocyte. Lymphocyte, these are also constituting 20 to 25% of total WBC. There are two major type of lymphocyte, that is B lymphocyte and T lymphocyte. Both B and T are responsible for the immune response in the body. So here we have seen these are the A granulocytes. Now coming to the platelets. Platelets are also called as the thrombocytes. These are the cell fragments produced by the megakaryocytes. These are the special cell in the bone marrow, and these normally blood contain this much platelets per millimeter cube. Platelets can release a variety of substance, most of which are involved in the coagulation of the clotting of the blood. Okay, so they are responsible for the blood clotting. So these are the cell fragments. Platelets are the cell fragments, and they are developed from the megakaryocytes of the bone marrow, and they release thromboplastin, which will initiate the clotting. Now the reduction in their number can lead to the clotting disorder, which will lead to the excessive loss of the blood from the body. So this way you can see that the coagulation is very important for this phenomena. The platelets are very important. Now coming to the blood group. Variety of the blood groups have been discovered. So the various type of groupings have been done for the blood. Two such grouping important that are widely used all over the world are ABO and RH. Now ABO blood grouping, was first discovered by Karl Landsteiner in 1901. He received the Nobel Prize in Physiology 1930 for this discovery. ABO blood types are also present in rodents and apes, including chimpanzee, bonobos and gorillas. Now ABO blood grouping is based on the presence or absence of two surface antigen. So these are what are the antigen? Antigens are the chemical that can induce any immune response. So because of the presence of the antigen, this antigen could be any protein, any chemical substance, any toxin, any parasite, any pathogen, and it will initiate the immune response in the body. So these are the surface antigen. Here when we are, these are the A and B surface antigen. So on the basis of this surface antigen, the ABO grouping has been done. So the surface of the erythrocyte contain a genetically determined assortment of antigen called agglutinogen. So these agglutinogen or the surface antigen, these are composed of glycoprotein and glycolipid. So based on the presence or the absence of various antigen, blood is categorized into the different groups. So within a given blood group, there may be 2 or more different blood types. As per International Society for Blood Transfusion, there are nearly 30 blood groups and more than 100 antigens have been detected on the surface of the RBCs. Antigen of other system are either very weak or function at low temperature only. That means that is why we are taking only these two grouping system into the consideration, that is the ABO blood grouping system. So here you can see this is the RBC structure. So this is the cross-sectional structure of the RBC, and these are the different blood groups that are present in the RBC. This is the ABO blood group and this is the RH that we will study. Now ABO blood grouping is based on the two glycolipid antigen called A and B. Genes for the ABO blood group are present on the chromosome number 9. Similarly, the plasma of the different individual contain two natural antibodies. So here you can see, first we will see the blood group on the basis of surface antigen A, B, AB and O. So this blood grouping is done on the basis of the presence or absence of the antigen. So if the antigen on the RBC is A, so the blood group is A. If the antigen is B, the blood group is B. If the antigen present in the blood group in the blood surface of RBC is A and B both, so blood group is AB. And if no antigen is present in the blood in the RBC surface, then it is the O blood group. Okay, so same way antibodies. Now we will see what are the antibodies. So distribution of antigen and antibodies in 4 groups. Here it is given. So these are the antibodies. They will be formed in response of the antigens. Okay. During blood transfusion, any blood cannot be used. So blood of a donor has to be carefully matched with the blood of a recipient before any blood transfusion to avoid the severe problem of the clumping. Okay, so from above mentioned table, it is evident that O blood group can be donated to a person with any other blood group, and hence O blood individual are called as universal donor because they don't have antigen. So when they don't have antigen, when they are given to any of the individual, they don't produce any type of antibody. So there is no reaction in the other individual. Okay, so AB individual can accept the blood from a person with AB as well as any other group of them. Therefore, such person is called as the universal recipient. So here we have seen that AB person, that is no antibodies are present in this. So it can take the blood from any of the blood group, that is AB, A, B, O, any can be give blood to the AB blood group. That is why they are called as universal recipient. So here also the same things have been given. Now after this, firstly we will see what is the RH grouping. So RH grouping, it was done by Karl Landsteiner and Alexander Weaver in 1937. So RH antigen similar to one present in the rhesus monkey, that is why the name RH is given, and these are also observed on the surface of RBCs of the majority of the human. So 80% of the human blood have the RH factor. So such individual which are having it, it is RH positive. Those which are not having it, it is RH negative. So most of the human population, 85% have RH positive, are RH positive, have the RH antigen, and very less are RH negative. Okay, so this generally these are. So next factor is here is the RH incompatibility. So that is also called as RH factor incompatibility or HbNB. So it is a special case of RH incompatibility observed between the RH negative blood of a pregnant mother with RH positive blood of the fetus. So what will happen, first time when it is exposed, RH antigen of the fetus do not get exposed to the RH negative blood of the mother in the first pregnancy. Okay, because as the two are well separated. Here you can see, though both are well separated. So there is no exposure of Rh positive and Rh negative. But what will happen in such cases? Mother, during the delivery, what will happen? Some blood will come from placenta, some blood will enter the in the mother's body. So what will happen? The antibodies against that Rh antigen will start preparing. So what will happen? So in the subsequent, when the anti-Rh antibodies have formed between the pregnancies, so during the second pregnancy, what will happen? The Rh antibody from the mother, they can leak into the blood of the fetus and destroy the fetal RBCs. So this could lead to the fatal to the fetus, and that can cause the severe anemia and jaundice to the baby. So this condition is also called as erythroblastosis fetalis, and this can be avoided by administrating anti-Rh antibody to the mother immediately after the delivery of the first birth. So first child. So what will happen if we will administer this anti-Rh antibodies? So what will happen? They will neutralize here. So after the neutralization, subsequent pregnancies and delivery, there will not be any of the issues. Here, Rh positive father, Rh negative mother. So Rh negative mother with the Rh positive baby. Now at the delivery, what will happen? Fetus will not be affected, no problem, but during the delivery, Rh positive baby blood will enter into the mother's stream. So what will happen? The antibodies will be formed. So Rh antibody will form. Rh antibody they will be remain in the bloodstream. So these antibody can attack the Rh positive baby's blood cells and cause the Rh disease. So here only after first between these pregnancy, if the anti-Rh antibodies will administer, they will be neutralized and there will not be any problem in the subsequent pregnancies. Now coagulation of the blood. Blood exhibit coagulation or clotting in response to the injury or trauma. There is a mechanism to prevent the excessive loss of the blood from the body. So dark reddish brown scum formed at the site of a cut or an injury over a period of time. We all have seen. This is called as clot or coagulum. These are formed by the network of threads called fibrin, in which the dead and the damaged cell formed of the formed elements of the blood are trapped. So this way the clot is formed. So the clot that is formed after any cut or injury, that will contain the dead cell or the damaged cell. Okay. So fibrin, these are formed by the conversion of inactive fibrinogen in plasma. So thrombin is involved here and it will activate the fibrinogen to fibrin. So thrombin in turn, these are subsequent present in the plasma, prothrombin. So prothrombin will form thrombin that will be activate the fibrinogen into fibrin. So this way the whole fibrin will form. So fibrin are the thread-like structure that will trap or that will seal the injury part. So the certain factor released at the tissue at the site of injury can also initiate the coagulation. Calcium ion play a very important role in the clotting. Now coming to the last part of this is the lymph. Lymph is also known as the tissue fluid. As the blood passes through the capillaries in the tissues, some water along with the many small water-soluble substances move out into the spaces between the cell of the tissues, leaving the large protein and most of the formed elements in the blood vessels. So these fluid that is released out is called interstitial fluid or the tissue fluids. So it has same mineral distribution as that in the plasma. Okay, so this tissue fluid or the lymph has the same mineral composition as that is present in the plasma. Now exchange of the nutrient gases between the blood and the cells always occur through this fluid. So that is very important function of this tissue fluid or the lymph. An elaborate network of vessels called lymphatic system collect this fluid and drain it into the major veins. The fluid present in the lymphatic system is called as lymph. The lymph is a colorless fluid containing specialized lymphocytes which are responsible for immune responses of the body. Lymph is also an important carrier for the nutrient and the hormones, and fats are absorbed through the lymph in the lacteals present in the intestinal villi. So this way we have seen that lymph is a very important body fluid, and it is going to play an important role in the immune responses of the body and the transportation of the nutrients from one place to other, one part to the other. Okay, thank you.